Hello, welcome everyone. In the introductory lectures you've heard that the immune system is an obstacle for transplantation, and in the previous lecture the history of transplantation and immunogenetics has been presented. However, in order to understand the role of the immune system in transplantation, it is essential to also understand its natural function. In this lecture we're going to look at the natural function of the immune system and what you need to know about it in relation to transplantation. The primary function of the immune system is to defend ourselves against different pathogens. Cells of the immune system develop in primary lymphoid organs like bone marrow and thymus, whereas most of the immunological action is taking place in secondary lymphoid organs, including spleen and lymph nodes. Upon injury or inflammation, immune cells also migrate to other peripheral organs, including the kidney. The immune system is a complex system, which can be divided into two parts, innate and adaptive immunity. The innate arm is the part we're born with. It is composed of, for instance, macrophages, granulocytes and K cells, but also includes complement factors and cytokines. Next to this, there's an adaptive arm, which develops during our life, based on exposure to different pathogens. This is formed by T cells, B cells and antibodies. You can already see that the immune system consists of both humoral components, so soluble proteins, and of cellular components, represented by the different white blood cells. An important aspect of our immune system is that it can very specifically recognize pathogens, and that it can discriminate between self and non-self. There is a central role for MHC proteins, in human called human leukocyte antigens or HLA. Within the groove of these molecules, small peptides are presented as the signature of such a pathogen and recognized by T cells of the adaptive immune system. In this case, both the presentation of peptides derived from bacteria and from viruses is illustrated. Recognition by T cells not only results in activation of these T cells and its effector functions, but also generates memory. As a consequence, upon a next encounter, a quicker and stronger response will occur. As you could see in the previous image, MHC molecules exist in two different forms, MHC class I and MHC class II. Although these molecules have similarities, there are several fundamental differences. MHC class I is expressed on all nucleated cells, consists of one variable chain together with the constant beta-2 microglobulin, and it presents peptides derived from intracellular proteins. These peptides have to be short, typically 9 amino acids, and the complex activates CD8 positive cytotoxic T cells. In contrast, MHC class II is only expressed on professional antigen-presenting cells like monocytes, macrophages, dendritic cells and B cells, and consists of two separate chains. It presents peptides derived from proteins taken up from the extracellular milieu, and these peptides can be variable in length, up to 25 amino acids. This complex activates CD4 positive helper T cells. In conclusion, MHC molecules, in combination with peptides, are essential to activate T cells in an antigen or pathogen-specific manner. We have learned in the previous lecture that the MHC genes are highly polymorphic, which means that in the population there is a large variety. Although this creates the transplantation barrier, as mentioned before, it has the advantage that within the population there is a great flexibility to deal with different pathogens, especially when pathogens mutate rapidly. In this example, you can see that different peptides from a viral protein can be presented by MHC molecules. However, for an individual person, the inability of its MHC proteins to present any of these dominant pathogen peptides will result in a sensitivity for this infection. Therefore, in an individual homozygous for HLA-A11, will not be able to respond to this viral protein. Since an individual has two copies of every HLA gene, this means that someone only has a limited set of MHC molecules. You might think that this has therefore only a limited capacity to present different peptides. However, both MHC class 1 and class 2 form a groove in which the specific peptide binding is determined by 2 or 3 anchor residues. The other residues of the peptide can be more variable, and hence a single type of MHC molecule can present a large variety of different peptides. The groove of the MHC class 1 molecule is closed at the ends, which explains why only peptides with a length of 9 amino acids can be presented. From the quiz, you've learned that the MHC molecules do not discriminate whether these peptides are self or non-self. This notion is important because it is a very commonly made mistake. So this lecture has given you an introduction on the immune system. We saw that the immune system defends ourselves against different pathogens and that the MHC molecules present small peptides of these pathogens, which are essential to activate T cells. We've also learned that an individual only has 2 copies of every MHC allele, whereas MHC genes are very polymorphic within the population. This variation in MHC is the explanation for the transplantation barrier. It prevents organ transplantation between genetically non-identical individuals, unless the immune system is suppressed, which will be presented in Module 3. In the next lecture, we will focus on one part of the adaptive immune system, namely the humoral immune response.